I'm Kara Haddock. I'm here at InMed with the Texas A&M Foundation. Today, you guys are going to be hearing from Daniel Powell and his partner, Navid. Navid, you have to pronounce your last name for me because I've been avoiding it for a day and a half. Khodaparast. They are going to speak to you guys on the innovation and their path and journey with their innovation over at Spark Biomedical. If you guys would please give them a round of applause and welcome them to the podium.
His last name intimidates everybody. It's totally, I had to work a long time to figure out how to say Khodaparast.
Thank you. 6 months.
Hey, thank you everybody for having us. I'm Daniel Powell. I'm the CEO of Spark Biomedical. This is Dr. Navid Khodaparast. He's our Chief Science Officer. We have a short little presentation here. I'm absolutely blown away with the program. What an amazing thing. I think I saw a counselor once when I went to A&M. The resources you have here are just something amazing. I do want to start off, if you remember one thing from the presentation, it's that we have amazing dogs. You can all vote who has the better dogs, not knowing which one belongs to us. I promise you won't. If it's the ones on the left. I'm going to turn that over. Navid will take us at the first.
Yeah, so again, thank you so much for having us today. The facility, the program that you guys are involved in is, I said the word unique earlier. It's really, truly an outstanding program you have at InMed. And, you know, my journey to become a founder of a company and an entrepreneur is a little bit kind of linear, but also kind of, you know, fragmented in a lot of ways. But I started my career kind of the same way many of you did. You kind of did a pre-med program as an undergraduate, whether that was, I know all of you are engineers. I was the standard biology pre-med. You know, I'm 90% of the class. I'm going to push hard. I'm going to go to medical school. I'm going to become a doctor, and I'm going to save the world. Part of that story was true. You know, Dan's heard it a lot, but you don't want to stand by me? Okay. You're so far away. But no, so I went to UT Dallas. I'm not an Aggie. I'm not a Longhorn. I'm kind of in the middle, but UT Dallas, we were the Comets, the whoosh. If you know anyone from UT Dallas, but biology pre-med, I did go to medical school. So the program I was introduced to and I was fascinated by it was not allopathic, but osteopathic medicine. So really looking at the body holistically and understanding how you could treat it with obviously pharmacologics, but also using the body's innate ability to heal itself. The school that I went to was short was PCOM, which stands for Philadelphia College of Osteopathic Medicine. Very prestigious school in Philadelphia, but they had a southern branch that I went to in Atlanta, Georgia. And I was, I think, the third class there. So some of you probably know what that feels like. The facility was not like this. It was new, it was nice, but I was really interested in clinical trials and research, and even preclinical research, which they didn't have. So I wanted to do a terminal degree in both DO and a PhD. And my focus was primarily in neuro. And after 2 years, you know, the hardest 2 years, I had a lot of conversations with advisors and friends, and my decision was to leave medicine and go pursue a PhD in something else. And I really didn't know it was going to be neuro at that time. And I saw a lot of treatment gaps in the neuro space, meaning a lot of my friends are neurologists, and I talked to them, it's like, do you cure anyone? And it's really, really rare for them to say, yes, I have done that. Neurological conditions, unfortunately, there's a lot of Band-Aids and not a lot of cures. And so I left med school after 2 years, and I returned back to my alma mater at UT Dallas. And I started it was an applied cognition program, which was kind of like an electoral program where you could go and you look at different labs and you would explore different applications of neuroscience. And there was, I was working at the Center for Brain Health at the time doing some fMRI work. And I was out the door, kind of like this kind of setting, and I was walking by, and there was a professor named Michael Kilgard that was giving a talk, and it was just the end of it. And I saw his last slide, and it says, "Vagus nerve stimulation cures all." And I go, what is that talk? Like the vagus nerve, come on, it controls the heart, the autonomic nervous system. What is this guy talking about? And I went in and I talked to him at the end of it. I was like, what is the vagus nerve? And he told me, well, why don't you come and take one of my classes? And I did. And that was my PhD advisor going forward. So Michael Kilgard, when I joined his lab, there was one goal, and that was translational neuroscience. I didn't want to do anything that was theoretical, everything, because he had a rodent lab, and everything that he was working on was vagus nerve stimulation. And the goal was to translate that to humans in your PhD. And so I shook his hand. I said, that's the goal. He was an auditory lab that we then transitioned into a motor lab. So we were using vagus nerve stimulation, direct vagus nerve stimulation, to activate neuroplasticity, reshape the cortex, enhance motor function. And at first, the first question was, can you even do this? And the second question is, we can, but how do you now apply it to different neurological conditions? And that led to my PhD work, which was in stroke recovery. And so we started the first animal models using vagus nerve stimulation to enhance plasticity, to regain motor function in rodents and rats. And then when I finished my PhD, which we promised that first day we had that handshake, was it would be translated to humans. And so my trajectory was a little bit atypical in the sense that a lot of times you do your postdoc at a different institute to kind of get more of a well-rounded education. But since my research translated directly to clinical trials, there was a company that was involved at the time called Microtransponder, which was a startup that was bred out of UT Dallas. And I was pretty much locked down in that. And I wanted to be. I wanted to see how the research went from animals to humans in a very short amount of time. And so I continued my postdoc for 2 years at UT Dallas while also working with a startup. And so I watched the work from animals go to humans and how the clinical trials got evolved. And that gave me that itch that I still have today, that entrepreneurial kick of answering really tough questions in a very short amount of time and being able to do it efficaciously and responsibly. And so my postdoc has then led me to numerous different startups, which is where I met Dan for the first time, which was a deep brain stimulation company where we're, essentially putting DBS electrodes and recording from the brain. And the best thing that ever happened to that company is that it failed, because that gave Dan and I the opportunity to work together and create Spark Biomedical, which we'll talk more about here shortly. How do I go to the next slide? Perfect. So I kind of talked a little bit about this, but I wanted to give a little background to some of the neuromodulatory approaches that I've been involved with, that a lot of people are involved with, and how that kind of evolved into what we call transcutaneous auricular neurostimulation. So on the left, you can see this is a standard picture that you'll find of what's called implantable vagus nerve stimulation. And so what you're seeing in the chest, they implant an IPG, which is an internal pulse stimulator or pulse generator, which then has a wire that will then be tunneled up to the vagus nerve. And then there's a simple cuff electrode that wraps around the vagus nerve and delivers mild stimulation, electrical stimulation to the nerve. VNS was initially FDA approved for epilepsy in the 90s. And later it was approved for depression recently. And then most recently, the work that I was describing about vagus nerve for stroke recovery, from that first implant I did till the day it was FDA approved, was 12 years. So I'm happy to say that now VNS is FDA approved for stroke recovery. But the problem with VNS, even though it's an amazing technology and it has a lot of use applications for treatment, is that it's invasive. It requires a surgery, I believe every 5 to 6 years, you have to go back in and have the battery replaced. So there are replacement surgeries that happen over time. Epilepsy is not a curable disease, so this is a treatment that you get every day. The device is turned on every 5 minutes for 30 seconds to help stimulate the brain and prevent the seizure. And so there's inherent issues with the technology. Auricular vagus nerve stimulation became a hot topic, I would say, around 2015, because a lot of the central dogma type clinical trials that were happening with VNS, vagus nerve stimulation, was being repeated with a non-invasive way of doing it. And so if you know the neuroanatomy, you have 2 vagus nerves in your neck next to the carotid artery, but both of these vagus nerves send a projection to your ear, which is called the auricular branch of the vagus nerve. And that specifically is shown in region 1 on the top right. And so that area is innervated by the auricular branch of the vagus nerve. Specifically, that dermatome area is called the cymba concha. And that's a highly targeted area if you want to activate the vagal network. And so that's one area that our company, Spark, targets right now with using transcutaneous auricular neurostimulation. The second area that we target, which makes our technology fairly unique, is that we're targeting 2 specific cranial nerve branches. One is region 1, which is the vagal component. Region 2, just in front of your ear, you can actually feel where the TMJ is, the temporal mandibular joint, and that's where the auricular temporal nerve resides. And that's a branch of your trigeminal nerve. And so by combining the stimulation of both region 1 and region 2, we've coined and trademarked this term TAN, which is transcutaneous auricular neurostimulation.
And I think we'd be remiss if we don't give a shout out. This original thought to do 2 different nerves was by our co-founder. He's got a PhD in neuroengineering. He was the one that should have been here, but he lives in Rome. So we're stepping in for him. But if he's in town, I'd love to bring him down here. But that was his vision, that we're going to go after 2 nerves and they're going to act synergistically.
Right. So this approach of neuromodulation TAN therapy is based on a lot of research, not only done by us, but also done by the field. And this is a diagram that I put together a while back. It's published now, showing kind of some of the theory and the hypothesis behind it, right? Looking at, by activating these 2 cranial nerves, the projections are being sent to the brain. And so you can see the auricular branch of the vagus nerve sends a projection to deep brain structures, specifically medullary regions of the brain and the brainstem, one being the NTS, which is the nucleus tractus solitarius. A highly studied area specifically for vagus nerve stimulation. And so when you look at a lot of imaging studies, you can see that not only when you have activation of the auricular branch of the vagus nerve, but you see activation in these brainstem region. The auricular temporal nerve also sends a projection to the trigeminal cervical complex, the TCC, which then sends signals up to the NTS. Regarding what Spark has been so focused on is opioid use disorder and addiction, we were very interested in how activating these nerves have a effect on the reward network. And so that's what you're kind of seeing in region 3, this kind of collection of different brain nuclei that kind of comprise the reward network. A lot of it is surrounded by the dopaminergic components that are provided by the ventral tegmental area. And then you have the nucleus accumbens, the amygdala, the prefrontal cortex. All of these are in concert together, helping you be aware of reward and being able to manage reward. So kind of going back to some of the neurostimulation, by activating these nerves, we believe that we can send a signal to the brain that releases endogenous opioids that fill vacant opioid receptors in the case that someone's going through opioid withdrawal and helping relieve their opioid withdrawal symptoms. So this is the device that we created and that we developed over the last 6 years now, and it's FDA cleared for patients, adults that are experiencing opioid withdrawal. And so the device is quite simple in the sense that it only has three components. On the top right is the earpiece. That earpiece is disposable, meaning that it's printed technology. There's no wires in it at all. So we use it's a dichromatic ink that essentially can be electrified and then provide stimulation via the hydrogels. So I like to think of it as kind of it's kind of an ECG pad sticker that instead of recording electrical activity, you're delivering electrical stimulation. And so each of those hydrogels is specifically placed over one of those cranial nerve branches. And so you got trigeminal, vagal, and then you have a return electrode behind the ear, allowing you to do biphasic neurostimulation. We have a cable then that connects it to a handheld controller. So you can see the device, the controller on the left with a screen on it. And that screen allows the patient to have a lot of autonomy with their treatment. A lot of patients with opioid use disorder feel like they're just told what to do, take this medicine, go see this counselor, do this, do this, do this. They don't ever feel like they have any self-awareness to their own treatment. And so we gave them a screen. And so they can change their neuromodulation. They can actually change the intensity to match their symptomatology. And so that's Sparrow Scent. It took a lot of engineering to come up with this, but it's quite a remarkable technology. For us to get FDA clearance, we did have to run quite a few clinical trials. This was our first study that we published in Bioelectronic Medicine a couple of years ago now. These were patients that were coming into a inpatient treatment facility. And so they all had opioid use disorder, and they were chronic users of opioids. I think they were averaging on average was 12 years of opioid use. And they were at a very high level of opioid usage as well. Some of these were about 1.2, this was heroin at the time, about 1.2 grams of heroin a day. So if you converted that to morphine equivalents, about 1,200 morphine equivalent, which would kill almost everyone in the room if you're naive to this. The treatment program was 5 days. So they would come in and they would go through withdrawal during 5 days and get medically managed. But we introduced them to the clinical trial in the sense that saying, well, instead of using a pharmacologic, like an opioid-based medication like Suboxone or buprenorphine, would you be willing to try neurostimulation as a way to manage your opioid withdrawal? And so patients consented into the trial. We had 31 patients that we enrolled in the study. It was a randomized control trial. And if you look at the graph on the right, this is showing the mean COWS score. And so COWS stands for the Clinical Opioid Withdrawal Scale. It's the gold standard. It is a clinically administered assessment. So a physician or a nurse would then assess the withdrawal symptom level of the patient at that particular time. And what you can see at baseline was that our patients had about a 15 on the COWS scale, which puts them in moderate withdrawal. And so they're sweating, they have GI issues, they're shaking, they have high irritability. They're showing a lot of what you would expect for someone going through withdrawal. And then the time markers at the bottom are essentially different times when we collected a COWS assessment. So for the FDA, they required us to show a 15% reduction, a 15% reduction on the COWS scale. And our primary endpoint time marker was 60 minutes. So within 60 minutes, we had to have a 15% reduction. What we saw with using neurostimulation, and no medication was provided during just this 60-minute time phase, is that we had about a 45% reduction in withdrawal. And so the FDA found this to be highly clinically meaningful and obviously safe because we had virtually no adverse events in the study. Following 60 minutes, patients were then allowed to have a comfort medication protocol. So this is essentially it's not an opioid treatment, opioid-based medication treatment. It was more using other medications like Zofran to help with nausea. And if they have pain, they can take Advil, Tylenol, and stuff like that. So they had ancillary medication for the remaining part of the study. There we go. And so we did continue to monitor the patients during their 5-day stay. We had 100% of patients sustain a clinically meaningful effect. By day 2, 84% had mild or no withdrawal symptoms within the first hour of treatment. So it was compelling for us to be able to. Yeah, of course. No, that's a great question. So the patients were directed to wear the device as long as they wanted to during the day. So on average, they were wearing it roughly about 14 to 16 hours. So mainly during their waking hours. So the control was the first 30 minutes. We're not showing that here. They were randomized into a group that had a sham device or an active device. The sham device looked and functioned just like the active device, but delivered no neurostimulation at all. And so when we looked at sham stimulation versus active stimulation just during that first 30 minutes, there was a statistically difference and showing that we had a superior benefit using the device. Yep, good question.
So sham and controls and wearable neurostim is really difficult. It's just so hard of setting, Yeah. Well, even that can activate the nerve.
Yeah. So another study that we became very interested was actually, Dan was at a neurotech conference and he became aware of a very vulnerable population that's exposed to opioids in utero. And these are babies that are born with what's called neonatal opioid withdrawal syndrome. And so there are moms out there that either are taking illicit opioids or they're taking opioids for pain management, or they're in recovery and they're having some type of opioid-based medication to sustain their recovery. And due to the presence of the opioid, it gets passed through the placenta and the baby is then exposed to the opiates. And so when the baby is born and they cut the umbilical cord, essentially the clock starts ticking. And not all babies will experience withdrawal, but about 60 to 70% of babies will have severe enough withdrawal symptoms that requires pharmacological intervention. And the drug that is typically prescribed for infants with NOWs is oral morphine. And so they deliver morphine at a rate that can help reduce the withdrawal symptoms. And then they slowly taper them off, usually over a few days or weeks. And so when we saw this opportunity, one, it was a big challenge to be able to get an IRB to approve this type of clinical trial. Because one, you're dealing with extremely vulnerable infants, and there's just not a lot of studies to show safety. We partnered with the Medical University of South Carolina for this trial because they were running a study already in infants using auricular neurostimulation to help babies that had global hypoxia during in utero. So they were trying to see if they could reverse any motor dysfunction to help them feed and prevent a GI tube. That was kind of the gist of that trial. And so I knew the researchers there fairly well, and I introduced them to the technology and said, hey, would you be interested in putting a grant together for the NIH to see if they would fund this type of study? And it was funded. And so we got a phase 1 clinical trial that was funded by NIDA to explore how neurostimulation can help reduce withdrawal symptoms in infants, and then be able to show that we could reduce the amount of time on oral morphine and also the time in the NICU. This was an open label study, so there was no control group, but we had 8 infants in this study, and this was published just a couple of years ago. The first thing that we were curious about is, do neonates even have the proper neural development that have the dermatome regions on the ear that can then send the signal to the brain and have the effect that we saw in adults? And the answer is yes, they do. There's actually a few light photomodulation studies that were looking at how signals being sent to the brain via laser acupuncture, as they called it. And so we targeted the exact same areas as we do in the adult, so the trigeminal nerve, the vagus nerve. And the way that this study was designed is every 3 hours is when they assess their withdrawal with a scale called the Finnegan scale. And every 3 hours is when they would give him a dose of morphine. And so based on the Finnegan scale, they'll be able to determine whether they can hold the morphine dose. If the Finnegan shows less withdrawal, then they can start weaning them off of the morphine. And you can see how they can titrate them over time. And so the way that we brought the technology in is at hour 2, so 1 hour prior to that 3-hour mark, we would deliver neurostimulation to hopefully by that time where they're checking the withdrawal symptoms would be less, thus requiring less morphine, and you get them off morphine faster. So being that this was a open label trial, our control group was the nation. And so the nation as a whole, if you look at all NICUs across the nation, it was 23 days was the average on oral morphine. The hospital that we treated them at MUSC in Charleston, South Carolina, was 16 days just for that one hospital. And the infants in our study says less than 9 days it was 7 days. So 7 days they were on oral morphine, and then they were out of the NICU within 11 days. And so this study went off for a number of different channels. One, it went to the FDA to help support what they gave us a breakthrough device designation, which is a kind of a more streamlined regulatory pathway that helps device and technology get to the FDA quicker. And then it also was submitted to back to NIDA to get a phase 2 clinical trial, which they funded, which will be ending this year. It'll be the 3rd year in this clinical trial, where we're targeting nearly 80 babies for that study. Your turn.
That's to me. So I mentioned I went to A&M. I got a business degree, and actually went to work up here in Plano at Electronic Data Systems. Well, no, that's in Dallas, up in Dallas. Ross Perot's old company, if anybody knows it. Didn't really know what I wanted to do. Was trying to figure it out. Went on, became a risk management consultant at KPMG. I did Y2K consulting, if anybody remembers that back in the day. It's when all of us convinced people that their computers were going to crash when the clock ticked over for Y2K. And then they spent tons of money on consultants to fix it. I did the .com, which if I don't know how many of you remember, that was the giant bubble that burst. That was when we used to get paid $20,000 to $30,000 per page on a website to develop back in the day. So it was a crazy ride. And then I had this great idea to open an internet game cafe. So you can tell I was like all over the place, trying to figure out what to do. I was very technical. I wired up all the computers. I did all the IT stuff. I kind of wanted to be a programmer for a while, even though I had a business degree, but I'm not a good programmer. And so I have this internet game cafe. So I decided to own a retail store. Anybody hear of GameWise? I think you're all too young for it. I was Mr. GameWise back in the day. And this engineer would come in, electrical engineer would come in at lunch and play World of Warcraft on my computers. And we started talking. His name was Ben Tranchina, and he was the director of electrical engineering at St. Jude Medical, which is now Abbott, and their neuro division right down the street. Long story short, owning internet game cafes is a great way to lose money, all of your money. And I needed to go get a job. And I got a software analyst job at St. Jude Neuro, in which I just fell in love with neuromodulation. I couldn't get enough of it. And it was sort of my breakout moment to really build a career. So I just kept getting promoted, which was fantastic. Kind of hit a point I wanted to do something else. So I went to Cyberonics, which is now LivaNova, which is implanted vagus nerve for epilepsy. And I ran global marketing. And then went to a startup to get back into DBS. I did DBS. I ended up running all upstream marketing at St. Jude Medical for DBS and SCS. Just had a passion for it. I love DBS. I love just this idea of putting these leads in the brain. And if you're in the operating room and the patient's shaking and their head's open, I'm standing behind the doctor, and you turn on the electricity and the patient stops shaking right there in the middle of surgery because they're awake during surgery. I mean, it's just fascinating. I'm having an allergic reaction to something. It's probably Houston, so forgive me. Anyway, went to a startup to get back into DBS, met Navid. We were not running it. It went under, but we were not running it. So that led to starting Spark Biomedical. And I had heard of this auricular stem, and I said it was horse crap because there's no way you could do that. You only could implant it. So then Navid schooled me. I was like, no, this does work. So the nice thing is I can call something horse crap and then be CEO of it later. I'm wrong a lot. But, in the 6 years, it's been an incredible journey. We did everything from product development, clinical trials, patent applications, FDA submissions, commercialization. Now we have reimbursement codes, audited books, FDA audits, DECRA International, ISO certifications, NIH grants. It's been an incredible journey, and it's just flown by, honestly. The hardest part about it is definitely commercialization, developing a product, going through clinical trials. Those, I know it's not fair to you to say those are the easy parts, but they're blocking and tackling, and you come to results. The unpredictability of market acceptance, changing buying habits has definitely been tough. But we built a great platform. And then along that way, we've continued to expand in the mental health area. So we're running additional studies for long-term addiction, alcohol use disorder, co-occurring opioid use disorder, and PTSD, all these things that exist together in the world of addiction and mental health. The other interesting thing, and I kind of want to take you on a journey of pivoting and changes, is we're launching a women's health initiative, or have launched. It's kind of in stealth mode right now. But it's how we got to women's health that is really interesting, and a series of journey experiences. So if you back up, the Feinstein Institute with Kevin Tracy reached out to us. They had developed this concept called the neural tourniquet, implanted vagus nerve stem, sending a signal to the spleen where your platelets are housed. The platelets are modified and express surface changes and essentially form quicker, stronger clots. What they showed in animal models was 50% reduction in blood loss. So they would do like a tail laceration or liver lobectomies on the pigs. And so this is really exciting. Like, okay, I can modulate blood and the platelets and reduce blood loss. What was really interesting was it's factor independent. And so they were able to take a hemophilia mice model and apply stimulation. And for 48 hours, that mouse clotted like a wild type mouse. So this was just fascinating. Hemophilia is the most expensive disease in the world to treat. It's about $300,000 a year to keep you healthy. And if you're unhealthy, the yearly cost immediately goes over $1 million. It is very expensive. So we thought, well, that makes sense. Let's tackle the single most expensive disease in the world with a $5 disposable earpiece. And then we started talking to hematologists, and they all said, we have this great new drug that's reducing the cost. It's called Hemlibra. Instead of having to put factors in 3 times a week, it's once a week or once every 2 weeks. We love it. I don't see a use for your product. And we're like, well, crud. Well, there's 40,000 Americans with hemophilia. There's 7,000 with hemophilia A. There's 7,000 with hemophilia B. And we're looking at hemophilia B, and we're like, it's not much of a business there. So we kept interviewing, and we ended up talking to Dr. Angela Wayand up in Michigan. And she specializes in women with bleeding disorders. Also, let me note, as you guys probably know, most hemophilia is 99% men. She says to us, why don't you consider women with bleeding disorders like von Willebrand's disease, where the majority of women have heavy menstrual bleeding? And we were like, this is very interesting, because you start to think of clinical trials and you're like, well, I got my control. I hate clinical trials that proves a negative event didn't happen. So let me explain that. It was in epilepsy. Did you not have a bad epilepsy seizure today? Well, I don't know. I don't know if I was going to. I don't know if the therapy did it. Bleed events are also, did you not hit your elbow and have a joint bleed today? Well, I don't know if I would have. But with heavy menstrual bleeding, we could see exactly the timing of the event. It was reliable. It was measurable. There's gold standard of measurements called the PBAC. And we started recruiting, found out it was really hard to recruit for, but we got the first 7, 8 subjects through, and the results were phenomenal. We saw about a 55% reduction in blood loss. So this is somebody who's losing a liter of blood over 6 days, and anemia and all the health problems for that. And we're reducing that by 55%. And we're sitting in a meeting, and then we're like, why aren't we just doing this for all women? And we started looking into this and started researching, and we're like, women's health is hugely overlooked in med tech. And the other things we saw in the study was significant reduction in cramps, significant improvement in mood, just all these other positive outcomes. And on our board of directors, we have an OBGYN, and he's like, when I was practicing, I would have given this to everybody. Like, why are we doing that? So we've rethought how we're going to do this. And so we're going to launch a whole new brand just for women's health. Our partner, Alejandro, thinks this can actually cure endometriosis. There's always a couple of hits that pop when you say that out loud. We got to prove it, but he thinks we can stop the migrating cells that migrate and don't heal. So really interesting and exciting. So this is kind of an example of, we thought we were going to, and we're going to look at surgical blood loss and everything, but we originally were over here looking at like dialysis port replacement surgeries, and we end up all the way over here in a women's health company. So just kind of sharing a journey of how you might have an idea that you have a hammer and you're looking for a nail, and sometimes you need to stop looking for a nail and like find the right solution or the right opportunity. 2 last slides, and then be happy to answer questions. One of the most amazing parts of this journey is having 3 founding partners that get along great. So Navid, Alejandro, and myself really pulling together as a team. When you go to raise money, when you go to have people believe in this, really having a solid team across the board behind you is important. And so, like I said, great founding partners, key to success. Even though I knew a lot about medical devices going into this, I'd been in medical devices for 15 years. There still was, I just went into this knowing I needed to learn a lot. I've never been a CEO before, so I Googled, what does a CEO do? I really did. I'll tell you what the answer is. It's set the vision for the company, onboard the team to execute the vision, and make sure there's money in the bank. These are 3 key things. The other thing that I've developed along the way is I believe in showing up, and it's why I'm here today. When I'm invited somewhere, or there's an event or there's a networking event, I just go and talk to people, and something magical happens all the time. It could be a future investor, could be an idea, could just be a new colleague that we're friends in this journey, and we can call on each other if we have a question about the industry, the FDA, whatever. The other thing, and it's part of having money in the bank, is if you ever do embark on a road like we've gone on, having a CFO, somebody who really understands the financial side, because that's probably going to be in your blind spot, is critical. And knowing that is, you're always up to speed, you're always informed at the financial health of the business correctly. Running out of money is not fun. We did it once. It's very painful, and it's the way companies die. I know that seems kind of obvious, but it can sneak up on you. And the last, I think, what is a quote that he reminded me to put in this is famous Mike Tyson quote, which is everybody has a plan until you're punched in the mouth. And then everything goes out the door and you start reacting. And that's business. We get punched in the mouth all the time, thrown, something from left field, and you just react, and, all problems are figure outable, I like to say. So thank you. I appreciate your time, and we're happy to take questions. I apologize for my throat. It's something got me.
Yeah. We'll actually start up with the question on Zoom. So the question is, what was the distribution of therapeutic choice by the study participants in terms of stimulation intensity, frequency, and duration?
Yeah, no, that's a great question. And so I assume for the adult study, so stimulation frequency pulse width, and the waveform were fixed for all patients. So the stimulation frequency on the vagus nerve was 15 hertz on the vagus, and then on the trigeminal was 100 hertz. Pulse width was 250 microseconds, and it was biphasic stim wave. The intensity varied across patient to patient because of the way that we adjusted their therapeutic intensity was based on perception. And so the going theory in terms of, you're activating a nerve if you feel it. So patients would typically go to a level that they can perceive, and then they would maintain it. But they were able to change it. So I think the average intensity across all 31 subjects for the vagus was 1.4. And then on the trigeminal was 1.0.
And it was milliamps constant current.
Yeah, milliamps.
Another question was, do we have a mechanistic understanding of how vagal nerve stimulation works?
Yeah, so for this population, it's kind of a two approach method in terms of the MOA. One is just based on the autonomics. So we all know the autonomic nervous system very well, sympathetic drive, parasympathetic drive. For patients that are experiencing opioid withdrawal, there is a very high sympathetic drive that's occurring during that state. And so activating the vagus nerve is classically known to promote parasympathetic activity. And so by pushing parasympathetics up, you can effectively reduce sympathetic tone and restore balance. So that's one mechanism that we think helps our patients. The second is what we're studying now in an imaging trial, specifically fMRI, where we think by activating the vagus and the trigeminal, you can release endogenous opioids. These are endorphins. And endorphins bind to the opioid receptors, thus alleviating the withdrawal symptoms.
I have two questions. First one is how close to the skin does the nerve have to, like, what's the penetration depth of that? Like you're providing electricity, just like, electrode, but reverse. How close it needs to be, that nerve, to the skin? Yeah, and I'll ask the second question later.
So the short answer, the closer the better. These nerves come very close to the skin. So you're about 2 millimeters away from the top of the epidermis to some of the subdermis tissues. You can drive electrical activation of nerves that are usually, you can go down even an inch. That's not a problem. But the problem with that is you're going to start running into impedance challenges, and you're also going to have a hard time controlling where the current is going. So for most transcutaneous approaches, it's usually within a few millimeters from the surface of the skin. If you want to drive activation to nerve structures beneath that, you typically would go with more percutaneous approaches or subdermal implantation.
Got it. And do people report to you as like they're feeling? Like, like you just said that they have to, that intensity changes. What do they say? Like that you have to? It
varies from person to person on that. Like the way I always describe it is kind of like this paresthesia tingling sensation. If you've ever worn a TENS device, like a muscle stimulator, you know that feeling. It can be kind of vibration feeling as well. Sometimes you can feel like your skin is vibrating, maybe a little muscle activation, motor activation that's occurring from the current spread. But yeah, it's mainly like kind of a tingling sensation.
Yeah, but like you said that some people had it like for 14 hours and they're like, so for 14 hours the device was on, they were feeling these feelings.
You forget about it. Your nervous system turns off, and you're like, and every once in a while, and we cycle it on and off for disruption.
Right.
It'll cycle off, and when it's coming back on, you're like, oh yeah, that's on me. But you really just forget it's there.
Okay. Yep. Cool. Thank you so much.
So thank you for the presentation. I thought it was very interesting that you mentioned about the spleen and how like the platelet activation changes. So it means that stimulating at the ear is also modifying parasympathetic output to target organs. So I was wondering if you had spoken to any cardiologists about this, because like first line heart failure therapy is neurohormonal blockade. And that's exactly what this sounds like.
Yeah, so I've never been involved. Well, I can't say that. I've done some work with vagus nerve stimulation for atrial fibrillation. I've never worked on heart failure, but there are a lot of implantable clinical trials using implantable VNS specifically for heart failure. Anthem, I believe.
Well, no, Anthem went out of business. So when I worked at Cyberonics, we had a group doing heart failure, and we're demonstrating that. And it's funny, we went to the right vagus nerve. I don't know.
More
why.
innervation to the heart.
Yeah, more
Yeah.
afferent. And they were showing that it was remodeling the heart and improving heart failure, but then they completely failed their pivotal. So I don't know I don't know if it's study design or whatnot, but
Yeah, they missed their primary endpoint.
they did really good when they did 40 patients in India. Everything was perfect. And then it's a joke, but you run clinical studies out of the US and it's like amazing results. And then you bring them to the US. Like a little more control.
I know you had mentioned that cramps or pain was a potential application of this. I was just wondering what the extent of that was, if you had considered it for labor pain or maybe pain management during outpatient procedures such as IUD insertion.
Right. So the going mechanism for cramp pain is essentially we're looking at what is it? It's called ischemic perfusion. And so as the muscles are contracting in the uterus, this type of pain is essentially due to lack of blood flow. It's one of the mechanisms. Vagus nerve stimulation helps promote parasympathetic activity, which also relaxes the smooth muscle walls on the veins, on the arteries. And so that increase in patency can help increase blood flow, thus reduce the pain associated with cramping. Regarding, labor and delivery, we haven't considered that. It's an idea that I think would work. But it's again, some of the OB-GYNs have been so focused when we talk about the utilization of this technology, they want us to go towards postpartum hemorrhage. And that's because it's a big problem. Even in the US, it's kind of surprising that we still have that as a problem. I usually think of third world countries when I hear that, but no, it's a problem here. And that's an area that they keep pushing us to go do more research on.
But we are doing a surgical pain study. We approached it a little bit more from opioid consumption. So we have a lumbar spine study at UT Southwestern where they're using stimulation perioperatively, and then we're seeing if we're reducing the overall pain from that procedure. So I think we've had one patient. It's moving slow.
Yeah, it's slow.
Yeah.
Slow roll.
So something weird, and it might sound like Navid's running 16 clinical studies simultaneously right now. Don't do that. It's bananas. Yeah. He's also a grant writing machine. So we keep getting funded, which is awesome, but we're not making it up when we're talking about all these studies. It's really abnormal. And you would say maybe you wouldn't have been running this many simultaneously.
One less would be preferred.
So anyway.
So my question is, what do we know about the long-term use of this technology? Do you anticipate it becoming something that the body gets used to, requiring higher intensity stimulation, or even, and this kind of sounds silly, but maybe it becoming less effective, or if it's as effective, becoming like habit forming or addictive itself? Just curious about long-term.
Fair question.
Yeah.
Everybody wonders about that. I'll give two examples and I'll turn it to you. Implanted vagus nerve stim for epilepsy, which is probably affecting all these functions, is continually efficacious. In fact, improves in efficaciousness over time. You get less and less seizures, and then it really is impressive. And so we have 30, 40, 50-year history of people implanted with that now. And then back to the fair question on, do you become addicted to stim? We've never witnessed anybody withdraw from the lack of stim.
Yeah, and I think, nerve accommodation, right? So kind of to your question earlier, you do perceive it less and less. The brain is very good at picking up a constant signal and turning it off. Like you put your shirt on, you don't feel your shirt anymore after a few minutes. It's the same idea. that's why you got to keep the brain aware. And so that's why we introduce duty cycles, kind of on and off patterns, so that it can't have as predictive of a nature to it. And that way the signal is always being presented and the brain is then receiving that signal as if it's, the first time it's seen it. That's really helpful. I think one of the negatives with our technology and as a whole with all wearable technologies is that it's hard for compliance for long-term use. And so for our studies, we rarely go past 30 days because it's harder for patients to comply with putting an earpiece on every day. Whereas with implantable systems, they have no choice, right? The system is on when it's told to be on, and it's on for the period of time. So that's one of the inherent kind of drawbacks to wearables.
I just had a question about when you said that you put the wearable on the patients, afterwards when you took it off, did the opioid symptoms, like withdrawal symptoms, return, or did it get better? Or do you have to keep the system on for the symptoms to not be there?
No, so the patients would stop at some point during the day, and we didn't see a return of the withdrawal symptoms, which tells us there was some halo effect. Either that we're providing so much endorphin into the brain that it's actually sitting in the cleft, and then that way when the signal is coming, the signal is stopped from the ear, they still have some supply of endorphins that they can use. But no, we even in the infants, we never saw escalation of withdrawal symptoms after cessation of stimulation.
So your device goes across a very wide range of diseases. Is it the same exact type of stimulation for all of them? Like, could you use the same device for treating bleeding versus pain, or is it a different type of stimulation?
The parameters are different. For example, for opioid withdrawal, we use lower frequencies, specifically on the vagus. For blood loss and hemostasis, we usually have a little bit higher frequencies on the auricular branch of the vagus. Again, these are kind of grandfathered in. There's not really been good dose response studies that show us this is exactly the parameter set that has the highest optimal therapeutic effect. We're just going off of 30, 40 years of vagus nerve stimulation and saying these are kind of the ranges of frequencies. And there's some literature that says lower frequencies may be better. So we kind of go towards that direction and we test it out. But even though we've been 50 years, dose response studies are hugely lacking. Whereas when you look at pharma, that's kind of where they start. And then they escalate into wider indications.
So then would you also allow users to tune their settings themselves to see what works best for them? Or is it set by
We don't let the users change the frequencies, the pulse widths. Physicians have the ability to do that if they think there's a rationale for it, or if they want to run their own kind of case study. But then when we do have another device just for researchers called Sparrow Link, and that gives them they can change any setting they want so that they can essentially customize the stimulation to their clinical trial. You guys should play with Sparrow Link. It'd be fun.
Do you
I think
anticipate?
they need that for the lab, I heard.
Yeah. Yeah. Happy to supply some.
Do you anticipate this being only in inpatient settings, or do you envision it being over the counter as well?
So that's a little bit driven by the FDA. So there's two questions in there. The FDA made this prescription, and you have to be under a doctor's care for opioid use disorder. For the women's health, we absolutely believe this should be an in-home, over-the-counter solution, and it's very possible. But this is approved for inpatient and outpatient use. So a lot of people do take it home. We actually sell, the majority of our sales are telehealth. Fun surprise, nobody wants to go to rehab, but they do want to get off opioids. And we realize this on the journey. So we actually have a full 50-state telehealth offering, and we ship this directly to people's house with a prescription. And that's been quite successful.
I was just going to ask if you guys have done any studies on what actually happens to the platelets from the increased vagal stimulation in spleen, because it has to be some sort of, we've all studied from like how clotting works, and it's very straightforward. Like, you know, there's a cascade of events that kind of link up together. So yeah, any progress on that?
Yeah, we just finished the first human study on that, where these are healthy human subjects that come in and we deliver 30 minutes of stimulation to the ear. And then we draw their blood at different time markers. So 10 minutes after stimulation, 60 minutes, and 2 hours after stimulation. We look at things like thrombin levels. We're looking at all of that from like efficacy and also safety reasons, because you don't want to create a prothrombotic environment, obviously. And then we do look at the platelet itself. And so we use technology called flow cytometry, which allows you to look at the cell surface protein structures. And what we saw, and just there's so many of them, but some of the ones that were interesting to us was specific lipoproteins. One was GP2B3A, which is responsible for increasing clot strength. And we saw a significant elevation just in that one marker. And then also looking at P-selectin. And so when you have the combination of increased glycoproteins, then we looked at thromboelastography, which kind of, there's a lot to that, you know, but it can tell you how strong your clot is and also how long it takes to form a clot. And we're seeing some really amazing effects specifically on clot strength with GP2B3A and also TEG max amplitude.
So have you guys thought about, like, if you had a patient that had like a defect, like Bernard Soulier or Glanzmann, like patients who would have defects in those, just it'd be cool.
So the dialysis port study that we're running, these people have a lot of comorbidities, and one specifically is acquired platelet dysfunction. And a lot of them are on medications like Plavix as well. And so this was, when we spoke to the vascular surgeon, my mind was, this is such a high risk population in terms of treatment effect that it's probably going to fail. And he was very convincing. And he essentially said, well, if it works in this patient population, then one, you're testing the boundaries of the treatment. But if it fails, it's also knowledgeable, because now you know it's kind of a mechanism. If it fails there, you know you're really dependent on the platelet.
And let's say that there's an NMed student that's like interested in, you know, exploring their own neurostimulation, I guess, idea that but they don't know where to start. What would be the first two, like one or two steps that, like, how do you get started on, like, yeah.
Well, yeah, just be friends with Navid. Yeah, just call me. No, I think we get a lot of researchers that just email us wanting, either they're very knowledgeable in neurostimulation and they know exactly what they want, they just need the technology, or they want to explore treatment parameters, or they want to explore options that could lead to a grant getting funded, or they want to introduce it to it in their own studies. And so me and my team, we take these emails and calls weekly to talk to researchers, and there's only a certain number of studies we can run, obviously. So yeah, we definitely encourage
6 teams are max. So
that's the max. So we encourage others to do it. Run more. Yeah.
All right. Well, thank you both for an excellent presentation. I think they both deserve a round of applause.
Thank you.